This paper investigates the mechanisms behind chain exchange in Diblock Copolymer Micelles, which are nanoscale assemblies formed by block copolymers in selective solvents. Understanding chain exchange, the process where polymer chains leave one micelle and integrate into another, is crucial for controlling micelle stability, responsiveness, and material properties relevant in drug delivery, nanomaterials, and self-assembled systems. Despite extensive studies, details of how individual polymer chains overcome energetic barriers to leave a micelle remain unclear, particularly regarding their conformation changes and free energy landscapes during escape. This work approaches the question by employing molecular dynamic simulations combined with advanced free energy calculations to map the chain escape process. The authors use collective variables related to the chain's configuration, such as junction point positions and end-to-end distances, to construct two-dimensional free energy surfaces and minimum free energy paths. They also implement kinetic Monte Carlo and forward flux sampling methods to estimate free energy barriers and escape rates under various conditions. By blending multiple computational strategies, the study aims to clarify the molecular pathways and energetic costs for chain exchange in Diblock Copolymer Micelles, providing a more detailed mechanistic understanding of this fundamental phenomenon. Figure 1 provides a visual example of a stable Diblock copolymer micelle as modeled in the study. This micelle consists of 36 polymer chains, each composed of 21 monomers of block A and 24 monomers of block B, held together by strong interaction parameters set at 25 kBt for like-like interactions and 23 kBt for unlike pairs. The micelle core forms predominantly from block A segments, while block B forms the corona, interfacing with the solvent. For clarity, solvent particles are omitted from the visualization. This stable micelle serves as the reference system to explore how individual chains begin their escape from such an assembly by probing their conformations and energetic states under these conditions. Figure 2 displays the computed two-dimensional free energy surfaces for two pairs of collective variables related to chain escape from the micelle, specifically RCM-JP and RE2E in part A, and RCM-CM and RE2E in part B. These variables respectively capture distances between the micelle's center of mass and the junction point or center of mass of the chain, along with the chain's core block end-to-end distance. The simulations were performed for polymers of length Na equals 15 and Nb equals 24, under interaction parameter beta delta epsilon equals 23. Each free energy surface is shifted so the minimum energy is 0, allowing ready visualization of energy barriers. Overlaid on each surface are minimum free energy paths, MFEPs, computed using the string method. The red lines trace these most probable escape pathways, and the yellow circles highlight the transition states, key configurations representing energy maxima along these pathways. The authors interpret these results as revealing how the chain conformation evolves as it moves away from the micelle core, and how free energy barriers depend on the collective variables. The similarity of the MFEPs on these two distinct variable sets further strengthens the mechanistic insights. This quantitative mapping sets the stage to understand the molecular steps of chain escape in energetic terms. Figure 3 presents the conditional probability distribution of the core block end-to-end distance as a function of the junction point's position, computed using the first free energy surface from Figure 2a. This distribution quantifies how likely various core block conformations are given different extents of chain displacement from the micelle. The red dashed line traces the mean end-to-end distance, and the yellow line shows the minimum free energy path. Visualizations illustrate four representative states: A, the chain fully inside the micelle; B, the chain extended into the surrounding solution; C, the chain collapsed at the micelle interface forming a globule; and D, the chain fully expelled into solution. The authors interpret this distribution as indicating that prior to fully escaping, a chain more likely adopts an extended conformation, stretching into the solvent. Near the transition state, the distribution becomes bimodal, reflecting coexistence of extended and collapsed states. This analysis supports previous findings where umbrella sampling showed growth of chain size, followed by collapse during escape, further elucidating the complex conformational changes chains undergo in this process. Figure 4 shows the minimum free energy path projected onto a single collective variable, RCM-XX, which captures the chain's position relative to the micelle center of mass along a spatial axis. This MFEP was extracted from the two-dimensional free energy surfaces shown in Figure 2. The authors present this one-dimensional reaction coordinate profile as a simplified way to understand the energetics along the escape pathway. The MFEP here summarizes the most probable path by which the chain moves from the micelle interior through to free solution, highlighting the energetic barriers and stable states encountered during the process. Figure 5 examines how the free energy barrier for a chain to escape from a micelle depends on the length of the core block, Na, by computing one-dimensional free energy profiles along the minimum free energy path. Part A presents these profiles for multiple core block lengths, all computed on similarly constructed two-dimensional free energy surfaces. As Na increases, the barrier height varies, reflecting how chain length influences escape energetics. In Part B, the corresponding free energy barriers extracted from both the MFEP and free energy projection methods are plotted versus Na on a log-log scale. The data fits a regression of the form ln beta delta F barrier equals A ln Na plus B, where the MFEP yields a scaling exponent very close to 2/3. This supports the Halper and Alexander mechanism, where the free energy barrier scales as the 2/3 power of core block length, consistent with a collapsed core escape. Interestingly, in the inset with linear axes, the barrier shows a roughly linear scaling at small Na values, indicating that short core blocks cannot collapse efficiently and incur different energetics. The authors compare their results with previous simulation studies, suggesting that their two-dimensional approach mitigates hysteresis and sampling issues seen in one-dimensional studies that obtained linear scaling. They propose that this improved resolution better captures the underlying physics governing chain escape through a combination of collapsed and extended conformations. Figure 6 depicts the Monte Carlo simulation setup used to study chain expulsion dynamics. The two bead spring chain models represent the same polymer chain at different stages. The left chain is localized at the interface between micelle core and corona, while the right chain has fully escaped and collapsed in solution. Dashed lines indicate virtual interfaces in the simulation that define reaction coordinate thresholds for forward flux sampling. These interfaces are strategically placed to quantify transition probabilities and free energy changes during the escape process. This setup enables investigation of chain escape kinetics and barrier heights under controlled order parameter progressions, bridging structural insights with reaction rate calculations. Figure 7 summarizes forward flux sampling, FFS results, using a collective variable based on the junction point's x-coordinate for micelles with varying core block lengths, Na, all normalized by the core end-to-end distance. In these simulations, the total chain length remains fixed at n equals 32, while Na varies. Panel A shows the cumulative free energy change along the escape coordinate, illustrating how the barrier builds as the chain progresses through virtual interfaces. Panel B plots the total free energy barrier derived from the cumulative free energies against Na, with error bars representing 95% confidence intervals from 5 independent simulations. Notably, the data follows a near-linear trend with core block length, indicated by a dashed linear fit line. The inset plots the corresponding rate constants, which decay exponentially with Na, consistent with expectancies when the core block does not collapse into a dense globule. The authors interpret this linear scaling as signifying that under these simulation conditions, the core block remains only slightly shrunk when exiting the micelle domain, possibly due to solvent penetration or dense corona effects. This interpretation aligns with experimental observations and contrasting mechanisms where core collapse does not dominate the escape process. Figure 8 presents results from the reactive trajectory ensemble method for chains with equal core and corona lengths, Na equals Nb equals 16, under conditions of z-1c equals epsilon equals 0.02. Panel A displays example escape trajectories showing how chains move along the reaction coordinate until detaching from the micelle interface. Panel B shows the conditional probability distribution of the x component of the core block's end-to-end distance, Re2Ex, as a function of the fraction coordinate def, which describes the progress of the chain escaping across the reaction coordinate. These plots indicate that chains primarily extend into solution, increasing both extension and fraction escaped, before ultimately folding and detaching. Panel C compares distributions of Re2Ex for chains localized at the interface, basin 1, and at the transition state just before detachment. Vertical dashed lines mark the mean values, highlighting that chains adopt significantly more extended conformations immediately prior to crossing the interface. The authors interpret these findings as indicating that the dominant escape mechanism involves chain extension rather than core collapse in these conditions. Combining with their enhanced sampling data, they propose that the minimum free energy path could cross over from a collapsed core escape mechanism to an extension-driven pathway, depending on core collapse propensity. In conclusion, this study elucidates the complex mechanisms governing chain exchange in Diblock Copolymer Micelles through a combination of molecular dynamics, free energy calculations, and kinetic simulations. The results reveal that the escape pathway of individual polymer chains involves nuanced conformational changes, with chains initially extending into the solvent before collapsing in certain conditions. The free energy barrier to escape scales with core block length in a manner consistent with the classical Halperin-Alexander theory when the core is collapsed, showing a 2/3 power law behavior. However, in regimes where core collapse is hindered, such as short chains or dense coronas, the barrier instead scales linearly with chain length, indicating an extension-dominated escape mechanism. This duality is captured by measuring multidimensional free energy surfaces and by forward flux sampling, highlighting the importance of collective variables describing chain conformation and position. These insights enhance our fundamental understanding of micelle dynamics, with significant implications for controlling self-assembly kinetics and designing responsive polymeric materials. The approaches presented also pave the way for further detailed studies of complex polymeric processes where conformation energy interplay determines function.